I'm Ben Fastow. I work for Ben Chen's lab here at UC Santa Cruz. We study mammalian neocortical development and olfactory bulb development. And I'm a fourth year student, so senior at UCSC undergrad. And I'm a neuroscience major. So the specific transcription factors we study, a lot of them have to do either possibly with Alzheimer's development, mental retardation, miscarriage, as well as other disorders such as autism now we're finding out. And it branches out, so the more we learn about the transcription factors we study now, we learn there's other transcription factors that are becoming novel, and it kind of progresses every year. We find out more stuff. Everyone has their own opinions on cortical development. So more so than arguing with other labs around the world, it's creating this cohesive model of what we truly as a scientific community understand about the developing brain. I was born in Israel, and then around the ages of 5, 6, I moved to America and grew up in Northern California. Both my parents were PhDs, but they're both engineers, not science related. And then I came into Santa Cruz starting as a bioengineering degree. And I kind of did the science classes you have to first before you go into like the circuitry and the engineering part, and I just really liked the science. The engineering wasn't for me, so I switched to neuroscience. And Ben Chen had an opening one summer while I was doing school, so I joined her lab and she took me in and started working pretty much end of my second year of college. I really like, the wet work, the in-lab activities. I feel like I learn a lot more reading papers that are coming out that are related to our research, arguing against our research or for our research. I learned more in my lab, I can say, than I probably learned in the majority of my classes in Santa Cruz. That's not because I'm like a bad student, but because when you're applying the material, it makes a lot more sense. So I learned, more about development of your brain from these stem cells into the developing, cortex than I did in any neuro class I've taken so far. I come in and in the morning I check all the work that we did yesterday, go over everything we did. Next I go and work with different staining, the different sections we need to stain, figure out kind of what we need to progress the project, and then I'll go on, take more pictures, analyze the pictures I just took on a microscope, and then go on tomorrow and based on those pictures and what we have, pretty much grab the new data, the new sections, restain those, which takes a couple of days, and then go on and repeat that process. So I would say, you're talking to all your lab mates on a day-to-day basis. I'm in constant contact with everyone. On top of that, we all have separate projects, but we work together cohesively. Our projects come in line into a main goal a lot of times. So I'm constantly helping other mentors and undergrads. In a lab or in different research experiences, you're going to get that the different side of learning that not many people have the opportunity to get. So I would definitely advise trying to get into the lab and doing wet work as much as you can in college.